You know, when you think about it, hunting for the genes that cause cancer is a lot like a police investigation. But imagine if you had hundreds, even thousands of suspects. That's exactly the challenge researchers are up against. Their list of potential genetic culprits has just exploded. So, how in the world do they find the real bad guys in a crowd of innocent bystanders? Let's dive in. Just get your head around this number. For only 3 common types of B-cell lymphoma, the scientific community has pointed the finger at over 850 different genes. 850! That is a massive, almost unmanageable number of leads for scientists to chase down. And that really is the million-dollar question, isn't it? It's the whole mystery we're trying to solve here. How do you sift through a list that gigantic to find the few genes that actually matter, the ones that could lead to new treatments and, hopefully, cures? So, how did we even get here? Well, it's kind of a classic case of having too much of a good thing. As our gene sequencing technology got incredibly cheap and fast, the amount of data we could generate just went through the roof. And that led to this flood of potential cancer genes, what some are calling a deluge of drivers. I mean, just look at the numbers for one single type of lymphoma, DLBCL. Back in 2011, scientists were working with a list of about 81 candidate genes. Fast forward just 10 years, and that number has skyrocketed to 680. That's not just progress, that's complete data overload. So what's causing this explosion? Well, a huge part of the problem is something called an orphan gene. Basically, it's a gene that gets flagged as suspicious in one study and then nothing. Crickets. No other studies can confirm the finding. It's like a hot tip that instantly goes cold, and it suggests that a lot of what's out there might just be noise, not a real signal. The researchers behind this whole audit, they put it very bluntly. They said the scientific literature right now is a total mess. It's a mix of real culprits and a bunch of irrelevant genes. And if we don't clean it up, these mistakes just keep getting passed on, which really muddies the waters for everyone. Okay, so why is this happening? What's behind all this confusion and all these conflicting results? Well, the problem really boils down to the evidence itself. This right here is pretty shocking when you see it laid out. In one major study, an incredible 82% of the genes they flagged were these orphans. They just never popped up again in other research. In another big study, it was 55%. It just goes to show you that different labs are coming up with wildly different lists of suspects. And the end result of all this is pretty clear, right? It creates this flawed, confusing map for cancer research. It's sending brilliant scientists down dead ends, wasting precious time, money, and energy chasing genes that might have absolutely nothing to do with lymphoma. But here's the good news. There is a way out of this mess. A team of researchers decided it was time to conduct a massive, systematic audit of all this data. They created a whole new framework to bring some order to the chaos by sorting genes based on the quality of the evidence. And what they came up with is this really elegant 3-tier system. So, Tier 1, that's your high-confidence list. These are the most wanted genes with strong, repeated evidence. Tier 2 is for the plausible candidates, the ones that need a bit more investigation. And Tier 3, that's for the outsiders, genes with weak evidence, a lot of which are probably just false alarms. And let me tell you, getting into Tier 1 is tough. A gene really has to prove its case. It either has to be confirmed as mutated in at least one other, totally independent study, or scientists have to prove in the lab that its mutation actually helps cause lymphoma. No more one-hit wonders allowed. So what happened when they took this super rigorous system and applied it to that enormous, messy list of suspects? Well, the result is nothing short of a dramatic shift, from total chaos to absolute clarity. Remember this number? That overwhelming, unreliable list of over 850 potential culprits? Well, after the audit, that number just plummeted. It's now 144. Think about that. From over 850 down to just 144 high-confidence genes. This is the new, reliable list of top suspects. Now, here's the breakdown of those 144 genes by the type of lymphoma. And while DLBCL has the biggest share, the really interesting thing here is the overlap. A lot of these core driver genes are actually shared between different lymphomas, which points to common biological weaknesses that we could potentially target with new drugs. So what does all this really mean? It means that for the first time, researchers have a clear, reliable roadmap. Instead of chasing 850 different ghosts, they can now focus their time and energy on the 144 targets that have the strongest evidence, which should, hopefully, speed up the path to finding new treatments. But, cleaning up the suspect list is really just the first step. It answers the question of who, but it doesn't answer how. And this has actually revealed an even deeper mystery, which is now the next frontier for researchers in this field. The challenge becomes crystal clear when you look at these numbers. We have 144 confirmed high-confidence driver genes. Awesome. But we only really understand what 58 of them actually do. That means for almost 2/3 of our top suspects, we don't know how they're causing cancer. The researchers call this the void of functional evidence. It's this massive gap in our understanding. We know these genes are involved, but for most of them, we don't know the specific mechanism they use. And that is the next great puzzle that has to be solved. The good news is, we have an incredible toolkit to start filling this void. Technologies like CRISPR let scientists go in and precisely edit genes just to see what happens. Other high-tech methods let them test thousands of genes at once. These are the tools that are going to help us figure out what each of these mystery genes is up to way faster than ever before. Which brings us right to our final thought. This amazing audit has taken a hopelessly cluttered, confusing map and made it clear. It's pointed us directly toward the most important targets. So the big question that's left is this. With this newfound clarity, how fast can we actually get to the destination? A deeper understanding of cancer, and ultimately much better treatments for lymphoma.